Is it the study of infectious agents that are not cellular, not alive, and are they ultramicroscopic? True or false? Are viruses cellular? No. Do y'all even remember in chapter 1 when we discussed the 6 organisms? And researchers argued, going, they can't do anything on their own unless they invade a cell. But because they were ultramicroscopic, they stuck them in there. Okay? So they are not cellular, they are not alive, but they are infectious particles. I have a question that says all of the following are viral characteristics except. So you've got a table that talks about characteristics of viruses. So I would learn that. You also have left justified stuff in your notes right after this section that talks about characteristics of viruses. So I've got 2 or 3 questions that said all the following are viral characteristics except. So make sure you go back and study those. Viruses, can they infect bacteria, protozoa, fungi, algae, plants, and animals? Obligate intracellular parasites of bacteria, protozoa, fungi, algae, plants, and animals. I'm telling y'all, my tests sometimes are verbatim. Just saying, that's verbatim. Let's see. Which of the following is not a true statement of viruses? Make sure that you know they are not cellular. In this, it says not cellular. Down here, notice that they can be double-stranded or single-stranded RNA or DNA too. So again, this chart, you've got to memorize that because I got another question on it. Are viruses very specific to what they can infect? Virus surface, very specific. So yeah, true. Again, know this thing. Viral replication. When viruses replicate inside a cell, do they normally kill a cell? Viruses can cause cells to burst open almost immediately after replication. Or the cell doesn't burst open, but it'll damage that cell in such a way that the cell will still die. So viruses kill the cell they're in, period. Are there more RNA family of viruses or DNA family of viruses? RNA. Is HIV an example of a retrovirus? If you go back and listen to Integrities, I talk about it. So we just learned about transcription where messenger RNA copies DNA. There are viruses that are RNA viruses, but DNA copies messenger RNA. HIV is one of those. And he's at the top of that little list. He's right there. Because right there's the word retroviridae. Is herpes an example of giant multinucleated cells? If you go back and listen to Integrity, I talk about those. Know the steps. Oh. And right there, there's the answer there too, multinucleated. There's the answer. Know the steps of replication. So viruses will attach. So then they go inside the cell, they start replicating, mature, and then they release. But know the names of the steps, know them in order, because I got a question on test talking about that. Down there in the box in the bottom, lysogenic with an IC is the same word as lysogeny with a Y. Know the definition of lysogeny, latent, and prophage. I have those on test. But viruses, can they get inside of a cell and stay there for lifelong? Yeah. HIV is one of those. Once you get it, you don't get rid of it. Herpes is one of those. So know the definition of latent. They just sit there, until they're triggered. There's a question with lysogeny right there. Are eggs used to cultivate viruses? True or false? What's great about egg is that you can see every growth stage of a vertebrate, and then you can test a vaccine on them. So yeah, perfect for that. You can even grow them in plates and see how they burst cells here. You can even see normal tissue versus tissues that have been infected by a virus there. So those are all interesting. Here's cytopathic effects. Know that those are no multinucleated inclusion bodies are examples of those. Here we also talk about mad cow disease. What's unique about it is we understand, we just covered chapter 9, how nucleic acids replicate, and these viruses are only made of prions, which is proteins. They don't know how they replicate, and that's the reason they're scary. And then they're associated with brain and spinal fluid type tissues, so you don't want to eat from these cattle. In the United States, for years, I could say we've never had any known cases, but we have since then. The human case for that is called the Crutchfeld-Jacob disease there. But now we're having to worry about deer. You know, bow season in Tennessee just opened this weekend, and I hunt with my dad all the time, but you see billboards on the side of the road now that won't let you take deer from one state to another because they're worried about the chronic wasting disease. And it's a cousin to mad cow disease. And notice what all is highlighted on that deer. So if you go to other countries where they have mad cow disease, you're not allowed to give blood over here in this country if you've lived somewhere else a long time. Or if you go on mission trips, you know, they'll say, oh, but it's custom, you gotta eat their food, blah, blah, blah. I wouldn't. I don't care if I insult them, if I don't know where it came from, because they will cook every part of an animal. So that brain and spinal fluid, they'll boil, sort of like we do chicken breast, and then that frothy stuff comes to the top. Same thing, they'll boil this brain and spinal cord, and then they eat that frothy stuff, and it's called cheesehead. The water that they boiled it in, like we would chicken broth, make soups, the base of some sort, they use that for soup. So even if you think you're avoiding the meat, you're gonna eat that, and that could have prions in it. So that's not good because they accumulate on the brain and spinal cord. The worst part, if you live in America, that we would come as close to eating it would be a T-bone steak because that comes off the spinal cord. But in deer, notice that's highlighted, same sort of an area. They say, my dad is really worried, he's like, you won't be able to deer hunt probably about 15 years if this takes over. Because, and then they're worried about that going from the animal to the human, and already know that it can cause neurological problems in humans if they contract it. So it's kind of a scary thing. And we do most of we eat deer meat more than I eat beef actually at my house, because we'll do that. So kind of scary, kind of things to think about. So there's that one. So I've already covered all of those. Know what TSE stands for. There's BSE at the top of that. TSE's there. There it is in parentheses. I'd make sure I knew that. Then I've got some questions from the end of the chapter, so make sure you still study those. Chapter 7, we have some stuff here. Know all these are factors that can affect growth of organisms. Know what extremophils are and what kinds of environments they can grow in. Know the definitions of all 4 of those words up there, macro, micro, and essential nutrients, because I've got questions that go with those. Know the definition of organic, or that means carbon and hydrogens are attached. Know that 97% of a dry cell of a weight is organic. Know all that. Know what TNOP stands for. That comes out of that bottom thing there. So, I mean, if you study my PowerPoint, you pretty much got it, right? Okay. Know the definition of autotrophs. There's TNOPs again. All of these are talking about autotrophs, heterotrophs, and saprotrophs. Saprotrophs are decomposers. So I give you several slides on autotrophs, heterotrophs, and saprotrophs. I would know these different ones here because I asked some questions on them. Then I see about 4 questions that go with my notes that are left justified. So make sure you're studying that left justified stuff from that. And then in your PowerPoint, This is matching on my test somewhere, this table, I'm always giving you prefixes and suffixes, so to me, that's freebies. You should know these, like, troph means food, feel means to love, ode means to live, hetero other, auto self. I've been saying that off and on all the time, so make sure you know that, to me, that's freebies. Then here's active and passive transport and tonicity. So know the definitions of active and passive, know the definition of diffusion and osmosis, and then iso, hypo, and hypertonic, because I got questions with those. Over here on the right, you got psychrophile, mesophile, thermophile, hyperthermophile. These are organisms that like different temperatures, so study that. Also know the definitions of minimum, optimum, and maximum, because I've got a couple of questions on that. Know definitions that relate to oxygen, anaerobic, aerobe, micro-aerophile. Know the terms that go with organisms that love acidic environments or basic environments. Know the definition of a halophile. Know all of these down here, because I've got questions asking about all these different types of relationships. And then you got pictures that go with those. Okay, then we move into binary fission. There's your picture of binary fission. Still looks much simpler than what we just covered in class, but no binary fission and the definition of generation time. That's how long it takes to go from one and double into two. Here's a growth curve, no lag. This is exponential, or it can be called log, stationary death phase. Understand that. And then then I got questions from the back of the book for that chapter. And that's the end of that one. Boom, boom. There he goes. So chapter 8, it's got some enzyme stuff in it, but first thing I've got a question on is metabolism. So know the definition of metabolism. Know all the characteristics of enzymes. So there's another table that you need to know. Table 8.1. Where's that one? Know these enzymes, catalase, oxidase, hexokinase. I've got a couple of questions of talking about different enzymes. You come back up here, kind of know the definition of anabolic, catabolic reactions. Here they are, too. There's where actually the definitions are. But know those. Know what an enzyme is, a coenzyme, and a cofactor are. So I got slides on those and questions on those. Another one with enzymes is this one. Know these enzymes, lactose, DNA polymerase. We just talked about those and know their function because you got a section where you match those. Know what competitive inhibition is. Competitive inhibition means every enzyme's got a certain function. Researchers know that, so when they make medicines, they'll make a mimic to the substrate. And when you're taking the medicine, hopefully the more medicine you get into your system, it increases the chances that those enzymes will collide with the mimic and it'll shut down that enzyme. So that's what you're seeing in that picture there, along with the one right after it. So this is competitive inhibition. This normal substrate fits it. They fit like a puzzle, and then the reaction can continue if you're on this side. But if this enzyme binds to a mimic, notice it comes here and it stops the reaction. Some know what an allosteric inhibition is. You can have an enzyme that will make all these products, then the product will go into a secondary site, closing off and shutting down that enzyme. I got a question asking that. I talk about different forms of energy, so know that. Know this slide, exergonic, endergonic. Know the structure of ATP. It's the same structure. If you cut off these other two phosphates, it's the same structure as adenine. That whole thing you saw with DNA. Know the definition of bioenergetics. Clear there. Know how many units of ATP is produced by glycolysis and the Krebs cycle down at the bottom. Here's the definition of glycolysis. There's your mitochondria's definition. Okay, then we move into another chapter. So definition, will you open? Probably gonna open 100 times then. Know the definition of genetic engineering, biotechnology, know the definition of semi-conservative replication, know complementary base pairing, semi-conservative replication, I just said that. Know pyrimidines and purines, because I have a lot of questions for my notes. So that's the reason I say take notes. So know those. Here's questions saying, in regards to the nucleotides, how many carbons are directly or integrated into that sugar portion? You're gonna say 4. Which one holds on to the phosphate group? The 5th one. Which one holds on to the nucleotide? The 1st one. What types of bonds hold the nucleotide to the sugar? Go back and look that up. What types of bonds hold the nucleotides together? Hydrogen. I asked that. Know how many bonds hold A's and T's together? 2. And then C's have 3. Know all your enzymes and what their functions are. That's on your test. And there's another one on semi-conservative replication. Know what replication forks are. Then I see a couple of questions here that come from the left, justified stuff. Know what a leading strand is, a lagging strand is, know what direction we like to copy. Know which types of RNAs do what jobs. Know about P and A sites on the ribosome, introns and exons. Then I've got questions from the back of the chapter. Then I've got the questions that we just went over under writing challenge. I'll open up. Here is biotechnology, know that definition, because I'll have that on there. There's your genetic engineering also, so know those. Know what a palindrome is. That's, palindrome's like reading the word race car forward and backwards. Same thing here, you have GAATTC at the bottom. If you read right to left, it's still GAATTC. And these are cutting enzymes. They like to cut in those spots. I'll ask questions about that. Some of those are left justified in chapter 10, know those. Know what gel electrophoresis is, which is this. So I have all that there, so make sure you know a little bit about that. Here's another picture of introns and exons and transcription, and here's like HIV reverse transcription. Or working in a lab, labs now realize what virus tricks can do, so they copy them. So here's another picture of your gel electrophoresis, so go back and listen to that. No questions from the back of that chapter. Know the definition of transfection, T-R-A-N-S, F-E-C-T-I-O-N, transfection. Know the definition of transgenic, T-R-A-N-S, G-E-N-I-C. And there you go. That's it. That's all it is.